So we had already discussed how viruses kind of lead what your book refers to as a borrowed life. So again, this is our discussion on viruses. And one thing we had gotten out of the textbook from yesterday was that viruses naturally lead a borrowed life. That means that they are requiring a host. In order to survive. Now, they require that host for a form of metabolism. That's how they get their energy. They require the host because the host can allow them to replicate or to propagate. So, without a host, viruses really cannot survive. We're going to talk about a certain type of virus called a bacteriophage, which is basically a virus that infects bacteria. So that's kind of the intro stuff that we really need to come into this lecture knowing. And I think we probably know most of that either from prior classes or the reading. Now let's talk about some history and structure of viruses. Viruses were long theorized to exist before they actually were proven to exist. And it was really hard to prove their existence because you can't grow a virus. You can only harbor a virus. You can only host a virus infection. But certainly you can't grow one. In other words, in a petri dish, a virus would just simply die. You have to have a host for that virus. And different viruses require different hosts, and that's kind of a unique matchup that's hard to get sometimes. So we could certainly say that viruses were detected indirectly. Long before they were actually seen. And your book goes into a little bit of the history of this. It talks about TMV, which is tobacco mosaic virus. And it also talks about some experiments by Wendell Stanley, which gave proof of viral existence. But again, a little difficult to do because viruses don't live on their own. Now, as far as their structure goes, we know viruses are certainly not cells. So what are they? What makes up a virus? Basically an outer protein coat called a capsid. And a lot of books just draw a bunch of little circles, and that's kind of supposed to designate that that's the protein capsid. No phospholipid bilayer here. Again, these aren't cells, so they don't need that requirement. And inside of it is some nucleic acid. Now that's a bare minimum for every virus. Each virus has to have at least those two components: the protein coat as well as some nucleic acid. Now, one thing to mention is that there are different forms of nucleic acids. So different viruses use different nucleic acids.
Sorry, Mr. Fields.
It happens. Some use double-stranded DNA. Some use single-stranded DNA. Some use double-stranded RNA. Some use single-stranded RNA, while others use, in a weird way, mRNA. So different viruses use different types. Now going back up to that protein coat, let's talk about that protein coat a little bit. Instead of calling it a protein coat, the other word that we use a lot is capsid. Which are made of subunits called capsomeres. Now, again, this is the bare minimum. There are sometimes other things that we could find around a virus. Some of those other things that we could find are called viral envelopes, which are basically something that would be found on the outside of a virus. The influenza virus, for instance, has a viral envelope around it. So we might want to add to this list of stuff that viruses have or are. We might want to add to this list is that there are viral envelopes sometimes. And these surround the protein coat, or the capsid. A good example of one that does this would be the influenza virus. Now, one thing I'll say is about this viral envelope is that a viral envelope is a lot of times, or actually most of the time, I guess, they're always derived from the host cell membrane. Now that means, if you think about it, that we have viruses, and there's their capsid. We drew on this out a while ago. And around that capsid is an envelope, a covering if you will. And that envelope is derived from the host cell's membrane. Now, why that? So my question to you, you can write this down. Why is the viral envelope derived from host cell membrane? And then also, what advantage does this give? So I'm going to give you like maybe 2 minutes to kind of just work through this and figure out why in the world does that happen and why would that be a bit if it ever. What are we thinking on the first one?
Maybe it bursts through the cell, so it picks up some of that.
Yeah, I think that's where I think that's kind of where we're going with that, is that since it is derived from the cell, it would only make sense that it would have the coat around it. Let me show you with a quick picture. Can you see this picture real quick? And this is just a simple virus. Do you see the virus right up there?
Mm-hmm.
It comes into the cell, right? Dispenses its bad nucleic acid, reprograms the cell. Now this cell's gone from being normal to being bad. And what do all bad cells do that have been infected with a virus? Not always.
Produce more viruses.
That's the best answer. They always, at the very least, they might kill themselves, but they definitely reproduce more viruses. Now think about it. As a new viral particle is being synthesized, which you're seeing right there, it's a self-assembly of the viral particle. Okay. Imagine it coming out and budding off the cell. What's it going to take around it as it buds out? That membrane. Now, so you can gather the answer to the first question. Why is the viral envelope derived from the host cell membrane? Because the viral particle buds off or buds from the host cell is when it's reproduced. Now, my question, my second one, which is more important, set up by the first one, is why is that valuable? Yes?
Resistance to the body's antibodies.
Maybe. Something a little bit better. Patrick?
Oh,
You first.
Yeah.
Haley?
So that it can just easily enter the cell, you know, like when they enter the cell and they're like whoosh, the membrane. You know what I'm talking about? I forgot what it's called.
Okay, I think you're going in the right direction.
It's not
Basically, we're agreeing that at the end, and I know that this doesn't have a viral envelope around it, so humor me a little bit and believe or work with me that this has a viral envelope around it. And you don't see one, do you? But if it did, it would be, what is this, like a light orangish color that would be around it. Now, that viral envelope is the same as the membrane of the host cell. So when this viral particle right here with its little envelope that matches this guy comes in contact with them, do you know what's going to happen? They're going to easily mesh. So it's for recognition. It makes for quick targeting of another host cell. So to answer those two questions, it says, why is the viral envelope derived from the host cell membrane? Well, because the virus buds from the host cell. You can think of it a little bit as basically a form of exocytosis. Now, what advantage does this give? Well, easy recognition. Now, that's at the very least what it gives, easy recognition. And there's so much more that's happening as well. Cool? Let's move on.
Hey, Mr. Fields,
Hmm?
what happens if this particular like bacteria that the phage is infecting has a cell wall?
It's not a problem.
It's not a problem. It'll just enter anyway.
Yep. Which makes those phages, I mean, it would make that a little bit more complicated, but still manageable. Let's look at the host range. We know what a host is, right? A host is the required organism for a virus. And the range is going to be the specific type of cell. So to kind of put these together, it's basically saying the host range is saying that viruses are specific to the cell that they can infect. A good example would be like the HIV virus can only target helper T cells. The HIV virus does not attack, though, a red blood cell, or an alpha cell of the pancreas, or a beta cell of the pancreas. Only helper T cells. Now, once it gets inside the host, we need to figure out what the host can do and what it's facilitating. So I wrote down hosting. How does a good host anything that's bad in your body? It buys you a little bit of time so that your immune system can learn how to fight the infection. So we certainly want to also remember too that virus particles really aren't, I don't want to say this awkwardly, but it's not that they're doing bad stuff. It's your body that's doing bad stuff in response to them being there. Now if they weren't there, your body wouldn't misbehave and you would feel well. But I want us to get that the viruses they're not releasing the chemicals that make you have a higher temperature. You're making yourself have a higher temperature. It's just your response. Now, another question people might have is, what type of machinery are we talking about here? Like, what does the host cell have to provide? What are the requirements to host a virus? And basically, it's any cell that has the normal machinery. As long as it fits in the host range, a host can host. But some of the things we're going to be using is, we're going to be using the host cell's enzymes, ribosomes, their tRNA, their amino acids. You're kind of getting a lot of protein synthesis right through here. Any free energy. ATP is the most global example of that. And so if you think about it, these are the requirements for cellular hijacking. Instead of carjacking, it's cell jacking. Okay. Are we cool with the way that stuff is? Do we have any questions about it? Okay, so here's what I'm going to do. I'm going to show you a video. It's a real quick little video that kind of just goes over